All right, welcome back. So now we're going to move on to chapter 10, which is biotechnology. This is a really fun chapter because there's a lot of cool stuff going on in science, and we're learning new stuff every day. We're getting new techniques, new tools, and this is just a really cool one. If, for someone who's in the sciences, every time there's like a new technique that's developed and it can broaden our field of knowledge, we get really excited. So I'm going to hit on some of the major things, and we'll talk about how we make a transgenic organism and a little bit about PCR, that you might have heard about the PCR test when it comes to COVID testing. So hopefully after this lecture, you might be a little bit more aware of some of the things that we do in science. So let's get going. So first off, how do you visualize DNA? Like we would have done if we were face-to-face, we would have extracted DNA from strawberries. There's a virtual lab where you're actually going to do that. You're going to extract it from like some cheek cells, and then you're going to run those cells through a PCR. But there's a protocol attached to where you can do it from strawberries, and it's what stuff you have at home: salt, dish soap, isopropyl alcohol, a coffee filter. It's really cool, and it actually looks like a snot bubble. It's actually really cool. So how do we see all DNA? So if you remember DNA, we're talking about nuclear. It's located in your nucleus and in that single cell. So for each of these little cells right here, there's 6 feet of DNA in each individual cell, which is fascinating. Now, with cells in general, when we look at them in the microscope, we have to stain, unless we're looking at chloroplast, because it's just qualities of how light microscope works. So if we want to stain to see certain organelles or DNA, we have to. This is a DAPI stain right here. So what it is showing is DNA. So that's why you see all the blue highlighted right there. We can also stain the chromosomes like we use for karyotypes that we talked about before. We can stain for microtubules if we want to look for stages of mitosis. There's all kinds of different things we can do. Now, we will commonly isolate DNA from cells and use it for different things, sometimes to generate GMOs, for cloning, that's one technique, and then for PCR and sequencing. Now, whenever we do this, we have to do several things. First off, we have to get the DNA out of the cell. So we have to break open the cells. Another term is lyse. Lyse open the cells. So we have to, kind of tear up that plasma membrane, start to get all the contents released, and then we pretty much wind up with the cell soup. So then we have to use a method of removing the enzymes that are bad, that commonly degrade DNA, because remember I talked about how RNA has a short lifespan. Well, also will target DNA, so we need to make sure those enzymes are not going. And then we also have to separate the DNA from all the cell contents that we just released, all the proteins. So we'll do this via centrifugation, and you'll do this in your little virtual lab. Now, one of the things we want to do is, because we're interested in seeing how these work, is maybe we can get cells to take up foreign DNA. This is called DNA transformation. This is a little bit extra. It's not included in your book, but it's a really cool technique where we can actually regulate and turn genes on and off by us doing it, not by how the cell regulates it. So if we want to understand how a certain gene functions, well, you want to see if it's turned on or turned off. How does it go through? One method that we do that is called this Cree-Lock system. It's really quite eloquent. What we do is we have our target gene, and we mark it, and we design it, and we put these things called lock sites between them. And then it's a mouse. We design this and we breed it and get it all set up. And it also has a GFP reporter, so we can look for fluorescence to see if it works. And then we have this Cree mouse right here, and this is a activator. And what the Cree activator does is it looks for these lock sites, and it's going to cut them out. So once we have our Cree mouse and our LOX mouse with our target gene designed, we'll have this Cree-Lox mouse. And what we do is we apply a certain drug, it activates the Cree, and it cuts out the gene. So we can literally make a knockout mouse. So if we want to knock out the gene in maybe a specific organ, we can do that. Like I said, it can be tissue specific, because sometimes some genes have multiple roles in different parts of the body. We actually do it to where we activate. It's really cool. So there's a lot of cool stuff that we can do in biology, and I actually used this technique a little bit in my research, because we did a knockout, and we want to make sure we only knocked out the certain gene in the testicle, because it also has a key role in the brain. Now, transgenic organisms are made all the time. One example are GMOs, but basically it's an organism that receives foreign DNA. So all kinds of different ones out there. If you go to the pet store and you see those fluorescent fish, most likely those are going to be a transgenic organism. We use a lot of them in science to help track like certain tissues and visualize them. This is examples of GFP mouse. But most of the time when we make them, we're trying to use it to study. We're not doing it for fun because these don't look as cool when it comes to running around in the cage. But a lot of the transgenic foods and GMOs that are made, it's in order to improve the shelf life, increase the yield. There's a reason behind it. But, there's a lot of pushback from it too, because then are we playing God and pushing to be what it's not supposed to be? So one important thing is once we insert the DNA, the foreign DNA, it is passed down to future generations. So it is permanently changed in that line. So let's make one. Now, here's an example where we're going to make a transgenic organism, but we're going to make a little bacterium right here. So here's a problem that is common, insulin. So insulin, it's a hormone that regulates the amount of glucose or sugar in your blood. And you have to have it for your body to function normally. And it's going to be produced by the pancreas. And the pancreas is a super important organ. I just dealt with about it. My dog having pancreatitis this week, and oh, poor little guy. Well, my carpets have paid the price. So what happens if your body doesn't produce enough insulin? You wind up with diabetes. So we have to have a way to make insulin. It just doesn't magically appear. So you're going to have to find a way for these people that are diabetic in order to inject it. So we have figured out a way to mass produce it is by using bacteria. So what we do is like a cut and paste technique. So we have a regular bacterium, and we're going to use these little circular pieces of DNA called plasmids, and I'll talk about them on the next slide. And we're going to use that to put in the human insulin gene. And we're going to insert that into this bacteria. And then the bacteria is going to make the insulin for us, and they just make a ton of it. Then we're going to go through the process of growing it, purifying it, and then bottling it for distribution. It's really quite cool. So how do we get the DNA for the insulin gene into the bacteria? So this is where we have to use a few things. So one of them is going to be called a restriction enzyme. Now what they are, I'd call them molecular scissors. They're made naturally in bacteria, and what they do is they are designed, and there's tons of different restriction enzymes shown by these little letters here. They target specific sequences. And when they do is they look for the specific sequence, and then they do a cut. They actually physically cut the DNA at the sequence, and it kind of creates it now into two ends. And we'll call these sticky ends normally in biology. So we have these sticky ends. So what we do is we look for ends, and when we design our gene that we want to insert, at the end of the gene, we make it to where it's complementary to the overhang or the sticky images. So ultimately what we want to do is cut the current plasmid open and then insert a gene to where it just matches up perfectly, and that's how we're going to get it inserted. Now our method to get the DNA into the bacteria, we use what's called a plasmid. Plasmids are small circular pieces of DNA, and bacteria love to share them with each other. These are separate from their nuclear chromosome. Just one little short snippet on that. And a lot of times when you see a lot of bacteria fighting antibiotic resistance, it's because they have it sharing it between plasmids. Now we're going to use these plasmids. This is an example of a plasmid map, and it has like all the different cut sites for the restriction enzymes and the location of how many base pairs between them. So these are super cool. I did a lot of work with these also. So let's show how we use it. So we have our gene of interest. This is our insulin gene right here, and here's our plasmid. Now what we're going to do, we know this restriction site what we want to go through and target. So we're going to the restriction enzyme, our foreign DNA that we have designed with the matching ends, and we're going to put it all in a tube. It requires a little bit of heat. So we put it in there, and then what happens is the restriction enzyme cuts the plasmid, and now our sticky ends that are corresponding with our gene for insulin match up. So it's like a cut and paste technique right there. So now we have our plasmids that have our specific gene in there. Now what we're going to do next is we're going to take these plasmids and put them in a population of cells, a bacterium, and then we have to add a few other things, it's salts and some other chemicals. And what we do is we kind of shock heat it, and what that does is put holes in the bacteria cell walls and membranes and allows them for it to take up these plasmids. Then we do a test later on, plate these bacteria, and we use this AMP-resistant gene to test if the bacteria took up the plasmids or not. So if they're white, they have the plasmids with the insert, but if they're blue, then we know they don't have it. So then we'll select the white colonies and we'll grow them up and grow more and more and more of them, and bacteria grow really rapidly, and then we can use those then to insulin. It's quite cool. One thing that we can also do is check to see if the bacteria took up our gene is do an analysis via what's called PCR. It's called polymerase chain reaction. PCR is just a short term for it. Now what PCR is making a copy of one gene a lot of times. It's just like pushing multiple copies over and over again. We use it if an organism might have our gene of interest, like if I was doing that mouse crossing I was talking before, it's called genotyping. So we want to make sure, yeah, okay, that's the right mouse, it has what it needs. So we'll use it. It's also used in DNA fingerprinting, so looking at crime scenes, you know, you have all these suspects, let's see if who did it. Now, it's very similar to DNA replication. You got to open it up, you got to attach it, grow it, and then you got to stop it. So this is a cheesy little PCR song that goes through it, and here's a video that kind of walks you through the steps so you can visually see it. But basically what happens is you have your original DNA. We're going to have primers and then the nucleotides and the DNA polymerase all put into a tube. So the first thing we do is we heat it up, which is the denaturation step. So high heat makes those hydrogen bonds, and then the two strands of DNA are separated. Next is going to be the annealing step. So we're going to cool it down, and what that does is it allows for the primers to attach. And then next is elongation. So this is we heat it up just a little bit, and this allows for the DNA polymerase to hop on and extend in sections. So after one round, we go from 1 to 2. And normally you'll have about 30 rounds that you'll go through. So now each of these two strands will go through the same process. How we get it to stop is we go through back to step 1 with the high denaturation. The high denaturation causes the polymerase to hop off, strands to split up, and repeat this over and over again. Now, after we've gone through the process of doing PCR, we have to go through electrophoresis. Now, what electrophoresis is when we take our DNA and we put it into an agarose gel, and then we attach it to a rig that puts an electric current and drives the DNA through the gel. Now, DNA has a negative charge, and we're going to use that to drive it from to the opposite charge. Depending on the size of the fragment will determine how far it's going to travel. So the larger fragments will not travel as far as the smaller fragments. Now, one thing that we do is we want to identify the size. So if I know my certain gene of interest is so many base pairs, I needed to compare it to a ladder. What a DNA ladder does is no number of base pairs. So no matter what, you know exactly, okay, 5 bands down, this is maybe 700, and this will always be consistent. And then you can actually go and visualize and see your different samples, how many base pairs each little segment is. It's actually really cool. You can use this commonly in DNA forensics. So, you know, you've heard about, I love to watch the crime shows, as I mentioned, they want to see if the DNA matched. So here's an example. Here's DNA sample from a crime scene, and we have 3 suspects. So it's just a matter of matching up. Do you know which one did it? So, you know, if it's like, okay, well, if you even just starting at this top band right here, you see this suspect 1 and 3 are missing it too. But you want to make sure that you have multiple point matches. Now this brings a question, what about identical twins? That is a great question. Do they have the same exact DNA? They are technically exact copies of each other, but this is where we'll look for certain differences, so maybe some SNPs later on. It also brings into a question, can your genome be used without you knowing it? With all this DNA sequencing going on right now, people are getting more and more information. The ancestry sites and all stuff. I don't know if it's highly regulated. The latest big ones, when they call it the Golden State serial killer, using information a relative have provided to a DNA ancestry site. So when you submit your information, what does the company do with it? Do they own your genes, which is kind of cool. Now, another weird thing about DNA forensics is kind of makes me think of Jurassic Park is, can we actually bring species back to life, extinct species? It's a debatable thing. Like I said, just because we can does not mean we should. Think about how much the environment has changed since then, and would we be able to manage this? So there's a lot of controversial things when it comes to this. Now, we are sequencing genomes like crazy. I know there's a lot of words here, just giving you a little bit more information. But basically what we're doing is we're taking DNA from a certain part of the body, and then we kind of go through and extract it, and we prepare this library that makes all these small fragments. And then these small fragments are run through another machine, and we make lots of copies, and we have to use computers to do this. And basically, this is just telling the direct order of sequences from start to finish. It makes it into lots of tiny little pieces, and then we have to use a computer in order to go through and piece it together, and we kind of look for similarities and overlaps. We're sequencing genomes like left and right now. It used to be super, super expensive and long too. The first genome, it took years to sequence the human genome, and now we are able to do things a lot faster. We're sequencing species like crazy, and sometimes we find that, oh, maybe I just read an article about giraffes. Here's two giraffes thought to be the same species, but there's enough differences to where they should be classified as two. So we're getting new things. It's really cool. And this kind of hints into DNA profiling. So this kind of goes with the crime scene too, also, because what they're going to look for is something called short tandem repeats. You have them in your genome. What these are sequences of nucleotides that are non-coding regions. So they don't code for anything, but what happens is, thanks to evolution, is you'll get a certain number of repeats, and it's thanks to your parents determining how many repeats you have. Now, why these repeats are important, because here's a statement up here: out of 1,000 nucleotides, there's only going to be a difference between you and a complete stranger. So think about, if you're out at the grocery store, hopefully you're wearing your mask and being safe. Think about a random person you come across. For every 1,000, there's only one difference in a nucleotide. So using these SDRs is one way we can actually amplify our DNA profiling to be more exact. So you can go through and see 3 different people. They all still have the same STR, but the number of repeats varies. So what we can do is look for different STRs, and then this is like use it with the crime scene DNA also, and see how many base pairs it is for each number of repeats. And this is another way that you can be more specific in mapping. So how do we do this? Once again, we extract the DNA and we make lots of copies. We're going to label the STRs with probes and put it through electrophoresis and then visualize it via profile. Now, one of the things is the more sequences tested, the more specific it is per person. Currently, the FBI recommends we do 13 STR sequences to test. Some of them in the UK only use 11, but they actually use a sex determiner to determine, make sure it's the right gender. Some of them include ones. It depends what country you are, but just a little bit more information about how they do DNA profiling. It's kind of cool. So this has been a really thing that's taken off, and it kind of scares me a little bit, to be honest. These DNA ancestry kits, yes, you do learn a little bit about yourself. You might learn, okay, I had some past relatives that might have lived in this area. So how it's done is they look for these things called SNPs, SNPs. Now, what they are is it's just a single nucleotide polymorphism that happens between different people. It's commonly seen like here, it's most likely in the non-coding area of DNA, and they look for this little snip right here. And what they do is they see between different populations, they're going to have the same. So what happens with these ancestry sites is they look for common SNPs that for each area. So whenever you submit your DNA, they look for these SNPs and then they match it up to these different databases. Now, the accuracy of these are kind of in limbo because, well, the more people submit, the more refined the areas will become. But it leads to that question, especially a bunch of them on this mental floss article, how do they use your information? You can also get some genetic testing from it, and it makes you question, okay, am I getting too much material? Are they telling you stuff that you probably don't want to know? Are you capable of handling? What if it's something bad? But this is another cool topic that also I bring up that Golden State serial killer again. It's, can the police or crime or the government actually use your genetic information with you? I don't know about the fine print. I haven't read it, but what if some discovery is made? You have some weird gene and you just send it off, you spit in your cup and send it off to find out, if you're Scandinavian or what. But what if they find something really interesting and then they make a lot of money off you? Do you get compensated? So it's cool. Check this out right here. You might learn a few things. Now, one thing I wanted to do is make sure you understand the difference between artificial selection and GMOs. This is what these next two slides are. So artificial selection, we've been using it in agriculture a lot, and this is just basically selective breeding. We're not inserting foreign DNA. We're just driving pretty much how evolution is happening. We're saying we're going to start breeding for this certain trait, and then eventually we get to the certain characteristic that we want. As I mentioned, in agriculture, this has been used a long time. All these different vegetables that you're probably used to seeing in the supermarket now are derivatives of the wild mustard. Dogs is another prime example. You think about their primary ancestor, which is the wolf, and think about how through selective breeding we got to all these different dog breeds. So this is where we are driving it. We're not going in the lab and altering the DNA, but we're this selective breeding. Altering the DNA is the GMO, or the transgenic organism. So anything that has incorporation of a gene from a different organism or a different genome is a transgenic organism or a GMO. Now, there's a controversy. You can be pro-GMO or anti-GMO. We had a discussion post on it. The reason I could see the positives behind it, we want to increase shelf life, yield, nutritional value. Think about where the vegetables are grown. Okay, so they have to be harvested and then cleaned and then transported, how long they sit on the grocery shelf and how long they sit in your fridge or on your shelf or your counter at home. You want to make sure they last for freshness for a long period of time. Yield, we only have so much arable land that we can use for agriculture, so the goal is to get the most product out for in the same amount of area, apply the least amount of pesticides. Can we make them more nutritious that we have? A lot of them make them resistant to pesticides, so we spray fewer chemicals, but now the insects figuring out, and now they're becoming stronger. So it's a win-lose situation, and you've seen a lot of push, especially for labeling of foods. Are they GMO free or anti-GMO free? Honestly, when my opinion is, money's going to talk. You can pay for organic and all the other stuff. It costs more. We've probably been eating these a long time and you don't even realize it. I don't have a problem with them, but it's all personal choice. And like I said, money talks. You will see that organic stuff does cost a little bit more money. Now, the last few slides kind of deal with stem cells and how we use this for gene therapy, and then we'll wrap it up with cloning. So stem cells are basically, they're undifferentiated cells. So early on in life, when we have that embryonic stem cell state, it's that inner cell mass that we are at the beginning of the zygote. And at that stage, the cells can become any type of cell possible. They're called totipotent. So at that point, they're like a blank slate. They have no idea what they're going to be. Now, depending on how they go through this thing called, or go through differentiation, and this is just a diagram called Waddington's landscape. So they'll go through, and as they get further and further down these hills and valleys, they become more and more differentiated and more committed. Now, when that happens, eventually they get to their final type of cell that they're going to be. Now, what's really cool with stem cell biology is we're now figured out how we can actually take it and go backwards. So maybe this is like a skin cell right here. And we can now undo the programming that happened. So we can take those genes that were turned off and turn them back on again and get back to this open slate, you know. Now, one of the things that we've noticed when we get to these cells that we brought back, we call them pluripotent stem cells, our iPSCs for short. They do have some marks on them. You know, the cell does have memory. And what happens is we then take the cell and then we can say, now I want you to go down this path. I want you to be a heart cell. You know, maybe we need to grow like some cardiac tissue to do for a patch. So you were a skin cell, and then I took you back, and then I made you into something else. Now, what happens is I mentioned these cells have been shown to have some memory. So, you know, one day he physically looks like a heart cell and is beating and all the other stuff. But then what if he wakes up one day and says, you know, I am having an identity crisis. This doesn't feel right. I used to be something else. Could it convert back to being a skin cell? That's one of the things we're trying to figure out. But this whole stem cell biology is really cool because it's patient specific. These are your cells. So maybe we can use them to treat certain diseases, transplants for organs, maybe, possible some organ tissues, all kinds of different things. One is the disease in a dish model. So maybe you have a certain type of brain disease, and there's all these possible drugs to use. Now, one way that you can do that is take like a biopsy of skin, boom, convert it into neurons, and then put that in a dish and then test all the drugs on the neurons in the dish and then see what works and doesn't work. And then based off the success from inside the dish, then you can apply that to you. So you're not testing a bunch of drugs on you, you kind of have a little bit of knowing like what's going to work and not going to work. Now, in stem cell biology, there's a huge controversy on the sources. Embryonic stem cells are the best, but in order to do that, we have to kill the embryo, and there's a lot of people against that. Versus the iPSC, they're not as pristine. They do have some of those cell marks and cell memory that we have to deal with. So this is a cool thing. It's controversy in between the field. I could talk for hours on stem cells, but I won't. But I do suggest you watch this. This is a cool video to where they talked about how they reprogrammed, and they actually took some skin cells and reprogrammed them into heart cells. And you can actually like see them beating in the dish. It's really cool. Now, one thing that we can use this to do is use it for gene therapy. So sometimes we have functional genes, defective ones. It just happens. There's two different approaches that we can do. There's germline gene therapy. I think CRISPR fits into this category, like I said right here. Basically, at the embryo stage, we're going to go through and fix it. Especially people that go through IVF and they know a genetic problem is probably going to happen. What they'll do is they'll have multiple embryos made, and then they will actually scan the embryos and look for genetic problems. Now, what could possibly be done is we could use CRISPR to probably fix that so you don't have to make as many. I had to read up a little bit more on the field and how it's changing, but this is fixing it at the early stage. Versus the somatic gene therapy, this is like, you know, just if it wasn't going to be fixed on you. Now, what somatic gene therapy does is it uses viruses to make functional proteins. And the only side effect is this is temporary. If you fix it at the germline level, permanent, you know, the entire, you know, child, if you're talking about humans, will come out and all cells in the body will be healthy. Versus somatic gene therapy, you know, we're only going to be, it's like a temporary fix, like putting a Band-Aid on and you keep having to go through it. But most of the time you'll use viruses that are going to have the correct gene that we need to have, and the virus is packaged into it, and then that goes and affects the cell. So it's a virus that's not going to make you super sick. It's a virus that's going to deliver the healthy gene into your cells, and that healthy gene will be then used to make the functional protein. So as I mentioned, this is only a temporary thing because this DNA will be eventually degraded. And finally, a little bit about cloning. I could talk about so many more techniques, but I had to restrict myself, and I'm going to wrap up with this one. Um, Cloning happens. It happens whenever we have identical twins, but we've been doing it for quite some time. I think frogs were probably some of the first ones that we achieved this through. The process is called somatic nuclear transfer. And we started, as I said, most likely in the xenopus or the frog first, but then we've gone through and done it in a lot more organisms. I heard of Dolly the sheep, but we've done it in a lot more in a bunch of different types of livestock. So what happens is we're going to have our cytoplasmic donor. So they're basically donating the egg, all right, the actual egg. So what we do is we suck out the nucleus. So we physically remove all the DNA by sucking out the nucleus. Then we have the sheep that we want to clone, or the animal we want to clone. So we take one of their mammary cells and we take the nucleus from that mammary cell and fuse it with the empty egg. So remember, this cell has no nucleus, so now we're going to fuse it with the cell that does have the nucleus. Now, once that happens, it kind of triggers the around the mitosis, and we get to the blastocyst, and then that will be implanted into the surrogate. So all DNA, that nuclear DNA, matches this donor. Any mitochondrial will be from this mother right here. Now this blastocyst is implanted, and eventually you will have an exact clone from the primary donor. So this is a really interesting process. Like I said, we've been doing it for a long time. Now, I do ask you to watch this video because we all love our pets and stuff. And this talks about cloning dogs. And there's actually a company that does it. And it's kind of makes you wonder how much you would pay. But it makes you think about what leads to the unique that makes you you, all the experiences, all the things that make up the personality. You know, just because you are physically and genetically a clone, are you the same thing? So watch this, it's really cool. And that is where I'm going to stop this one. Here's some review videos about biotech. There were videos scattered throughout, but this is a fun chapter. I hope you enjoyed it.